Welcome to the Cannabis Plant, a Clinical Perspective, Phytocannabinoids Learning section. The cannabis plant contains over 500 known active ingredients and is comprised of cannabinoids, terpenes, glycoproteins, and alkaloids. Despite its reputation, the majority of the active ingredients found within the cannabis plant are non-euphoric, and it is theorized that there are several active ingredients within the plant that remain unidentified. A cannabinoid is any compound which interacts with cannabinoid receptors. There are three main classes of cannabinoids. The endocannabinoids, as discussed in the endocannabinoid system learning module, are found in the human body and are natural endogenous compounds that interact with cannabinoid receptors in the body. Phytocannabinoids are the cannabinoids found in plants, and you may be surprised to learn that cannabis is not the only plant which contains phytocannabinoids. They are also found in echinacea, liverwort, and black truffles, as well as in spices such as clove and black pepper, among others. Most botanists believe that phytocannabinoids were formed by plants as toxins to deter pathogens and herbivores. The final class of cannabinoids are the synthetic cannabinoids, which are man-made, lab-developed compounds designed to interact with cannabinoid receptors as either a diagnostic mapping tool or as potential drug development compounds. The cannabinoids present in raw cannabis plants are not bioavailable to the human body. Instead, they are present in an acid form which requires decarboxylation to convert the cannabinoid into an active form which can then be utilized by the human body. Decarboxylation is the process of removing a carbon dioxide molecule which activates the cannabinoid into an active form. Decarboxylation occurs naturally over time as part of the drying and curing process of the cannabis plant, but it can also be expedited with the use of heat. This is the reason that the main method of cannabis consumption throughout the ages has been through smoking, as the direct heat applied to the plant allows for the almost immediate activation and delivery of active cannabinoids to the body. The most abundant phytocannabinoids found in cannabis are listed here. Most phytocannabinoids share common structural features that include a dibenzopyrin ring and a hydrophobic alkyl chain, which makes them insoluble in water and highly lipophilic. Contrary to other naturally occurring drugs such as opioids, nicotine, cocaine, or caffeine, cannabinoids do not contain nitrogen and hence are not considered alkaloids. Cannabigerol, or CBG, is the precursor to most of the cannabinoids produced by cannabis. CBG is formed in epidermal glandular trichomes by the condensation of a terpenoid and a phenol compound. CBG is then converted through synthase enzymes into the various other cannabinoid compounds. The amount of each cannabinoid produced by a specific cultivar of cannabis is determined by the volume of the synthase enzyme each plant produces. If a plant produces large amounts of THC synthase, that plant will have a large amount of THC present. If the plant produces a large amount of CBD synthase, the plant will have a large amount of CBD present. The production of these enzymes can be manipulated through genetics to produce plants that generate a predictable amount of each cannabinoid. Finally, the cannabinoid CBN is formed not by synthase enzymes, but through the degradation of THC after prolonged storage or exposure to light and heat. Delta-9 THC is one of the most prevalent cannabinoids produced by the cannabis plant. Delta-9 THC is a partial agonist of both cannabinoid 1 and cannabinoid 2 receptors. It is also an agonist of G-protein-coupled receptors 18 and 55 and vanilloid 2, 3, and 4 receptors, as well as acting as an allosteric modulator of glycine and opioid receptors. The agonization and modulation of these receptors contribute to the role THC can play as an analgesic and anti-inflammatory agent. Delta 9 THC is also an agonist at serotonin 3A receptors, which contributes to its role as an antiemetic. In studies, Delta 9 THC has been shown to be a powerful analgesic. It has also been shown to be a strong anti-inflammatory compound with 20 times the activity of aspirin and 2 times the activity of hydrocortisone. Delta 9 THC also shows significant clinical activity as a bronchodilatory agent, an antispasmodic, a neuroprotective antioxidant, and as an antiemetic and orexigenic agent. Cannabidiol, or CBD, unlike other cannabinoids, does not bind directly to cannabinoid 1 or 2 receptors in the body. Instead, it acts as a weak cannabinoid receptor antagonist and a negative allosteric modulator to help increase endogenous levels of endocannabinoids. CBD also acts as an antagonist at G protein-coupled receptors 18 and 55, as well as serotonin 3A receptors, and acts as an agonist at adenosine and serotonin 1A receptors, which has clinical impact for CBD as an anti-anxiety agent. CBD also inhibits cellular uptake of anandamide and mitigates the clinical effects of THC in the body. Interestingly, new preclinical evidence shows that CBD antagonizes tissue necrosis factor alpha, which points to a promising role of CBD as a potential anti-cancer agent. CBD has shown a wide range of clinical efficacy in studies. It shows activity as an anti-inflammatory, analgesic, anti-anxiety, anti-tumor, immunosuppressant, neuroprotectant, antioxidant, anticonvulsant, mood regulator, and has been shown to be cytotoxic in certain cancer cell lines. THCV, or tetrahydrocannabivarin, is the propyl analog of Delta 9 THC. THCV is a cannabinoid 1 receptor antagonist at low doses, but at high doses acts as a cannabinoid 1 receptor agonist, an interesting biphasic activity. At low doses, THCV has shown to produce weight loss, decreased body fat, and decreased leptin with increased energy expenditure in preclinical studies. It has also been clinically shown to be active as an anticonvulsant, an anti-inflammatory, and to increase insulin sensitivity and reduce blood sugar in preclinical studies. CBC, or cannabichromene, is the third most common cannabinoid after THC and CBD. Like CBD, CBC inhibits anandamide uptake and is a potent TRPA1 agonist. CBC also has a potent anti-inflammatory and analgesic effect, although less potent than THC. Like CBD, CBC also reduces the effect of THC intoxication in the body. CBC has also been shown to have some antibiotic and antifungal effect, as well as anti-cancer activity and antidepressant activity in preclinical studies. As mentioned, CBG, or cannabigerol, is the precursor to THC, CBD, and CBC formation, among others, within the cannabis plant. Although CBGA is not present in large concentrations in cannabis, there has been significant interest into the potency and activity of CBG as an individual cannabinoid. CBG inhibits anandamide uptake and acts as a potent agonist of the alpha-2 adrenergic receptor and TRPA1 receptors, which are correlated with its role as a potential analgesic. CBG is also an antagonist at serotonin 1A and TRPM8 receptors, which point to a potential role for CBG in the treatment of prostate cancers. Some unique activities of CBG are its antihypertensive effect, its powerful activity against methicillin-resistant Staphylococcus aureus, its activity against certain kinds of breast cancer, and its unique ability to inhibit keratinocyte proliferation, which points to a potential role for CBG in the treatment of psoriatic skin disorders. As mentioned, CBN, or cannabinol, is not formed from CBG like the other cannabinoids, but is instead an oxidized metabolite of THC. CBN is a weak agonist at cannabinoid 1 and 2 receptors, is a potent TRPA1 agonist, and is an antagonist at the TRPM8 receptor. CBN is highly sedating and has also shown clinical activity as an anticonvulsant, anti-inflammatory, and antibiotic. Similar to CBG, CBN inhibits keratinocyte production. CBN has also been shown to stimulate bone formation, and research is currently being done on the potential use of CBN as an osteoporosis prevention and treatment agent. We have only just begun to establish a clinical understanding of how each of the cannabinoids produced by the cannabis plant works within the body and the potential for their use in the treatment of various diseases. This chart summarizes the potential therapeutic opportunities of the non-euphoric cannabinoids found in cannabis. As indicated by the sheer number of opportunities listed on this slide, the potential clinical uses of phytocannabinoids are seemingly endless and represent a very interesting area of study.